Morning. We stopped here in the at the end of the first video, and we are going to continue explanation of the functional areas in the brain. Last time we were talking about the fifth lobe of the brain, or the insula, and we said that the insula is deeper than the rest of the lobes, and you can see it only by retracting the frontal lobe and the temporal lobe. The insula contains mainly the gustatory cortex, or the taste area, that receives impulses of taste sensation, which are sensory impulses, of course, as we're going to see on the next slides or the following slides. We also can see the olfactory cortex, or the olfactory area. And actually, the olfactory area is not part of the insula, but it's very close to the insula because it is on the medial surface of the superior temporal gyrus. And when I say the medial surface, I mean the surface that is inside, that is towards the middle line of the brain, that is deeper, that is towards the hippocampus, that we cannot see here. And then we have the auditory area. We are just going over the functional areas, or going over examples of the functional areas. The auditory area, as we said before, is present in the superior temporal gyrus, in the anteriormost part, and behind it we have the auditory association area. And we talked about them in the previous video. And then we have the somatomotor association area. We talked about the somatosensory association area in the parietal lobe last time, which is responsible for processing and integration. We say integration first and then processing, because when you integrate data, then you are gathering data, you are collecting data, and then you start processing these data, seeing what data have relations to each other, what data should be filtered, which data are more important than others. This is the function of the somatic sensory association area, in addition, of course, to understanding the various sensations that reach the sensory area that lies just posterior to the central sulcus. Concerning the somatomotor association area, it's in front or anterior to the motor area. Sorry. As you can see, the motor area lies just anterior to the central sulcus. It's this red strip of brain material, and it lies in the frontal lobe, most posteriorly. And in front of it, we have the somatic motor association area. Here, there is no understanding because we are not talking about a sensory area. So what does an association area when it comes to a motor area? An association area, when we're talking about a motor area, I mean a motor association area is responsible for higher planning of the motor movements according to the sensory data that we receive, meaning that this somatic motor association area will plan for and will decide which motor actions to be done. So if I ever ask you about the function of the somatomotor association area, it's planning for and deciding what motor actions to be done, coordinating these motor actions together, and at the same time making them relevant to the sensory information that the brain has received, meaning that the somatic motor association area cannot make a decision without previous reception and perception of sensory data received from the sensory area. So they are connected to each other. This is an evidence that the sensory area and the motor area are connected to each other. And now on the following slides, we're going to discuss functions of the different lobes of the brain. So if I ever ask you about the functions of the frontal lobe, you would say these two broad functions, controlling higher complex functions in human beings. And of course, we are talking about human beings because it's human biology, but this applies also to other mammals. and controlling speech. Okay, so these are the two main functions, and then there are details under each title. But what I want to emphasize is that the same answer, the opposite of the answer, actually will be answered if I ask you about lesions in the frontal lobe. So concerning that slide, you might be asked about the functions of the frontal lobe, and this would be a straightforward question, or about what happens when there is a lesion in the frontal lobe. I might also say what happens if there is if the frontal lobe is affected by a brain tumor, a benign tumor or malignant tumor encroaching on the cells and damaging the cells, or what happens if the frontal lobe is affected by a stroke, for example. So what are the higher complex functions that the frontal lobe controls? The higher, most abstract functions, which are your personality, your behavior, your emotions. You might be amazed to find emotions here, because we talked about emotions in the limbic system when we were answering the questions of the comprehension. Just like memory is controlled by several centers in the brain, emotions are also controlled by several centers in the brain, and they are all interrelated. But every lobe will deal with emotions according to its specialty. So, for example, the frontal lobe is always um, uh, related to judgment, to decision making. So, this is where you will have like the brakes for your emotions that arise in the limbic system. So the limbic system controls your emotions, but the frontal lobe has a higher control on your on the limbic lobe and on your emotions. And if you use the frontal lobe properly by training, by learning this, you can control your emotions. It also controls behavior. We have talked before about the amygdala, and a removal of the amygdala makes the person aggressive, or the experimental animal actually aggressive. But here the frontal lobe is also responsible for behavior. So people uh, who suffer from brain tumors can have changes in their behavior, changes in their personality, maybe more aggressive behavior, or maybe the opposite. They might be depressed. It also controls the habits of motor actions, like making coffee, making tea, driving a car, and so on. Judgment, of course, planning, problem solving, decision making, and all this is related to intelligence. But intelligence is a broad word, because we'll be learning that there is social intelligence, there is mathematical intelligence, and so on. Focusing, of course, self-awareness, but I repeat again and again that self-awareness, for example, is not only a function of the frontal lobe, but it's also a function of the parietal lobe, as we said before, because we said that if you have a lesion in the sensory area in the parietal lobe, you might neglect a whole part of your body. Sequencing and organization. Sequencing and organization have been seen in the parietal lobe and in the frontal lobe. But in the parietal lobe, um, this is different from the frontal lobe, because in the frontal lobe, sequencing and organization is mainly related to the complex motor actions, whereas in the parietal lobe, sequencing and organization is different. It is related, for example, to solving math problems and different other aspects of sequencing and organization. But overall, both lobes, the frontal lobe and the parietal lobe, play a role in sequencing and organization. The frontal lobe, mainly related to motor actions, will organize and decide the complex motor actions that you do. Of course, it controls speech because it is related to Broca's area, and Broca's area controls and coordinates the small articulator muscles around the lips, in the tongue, in the pharynx, around the larynx. So any damage to Broca's area will result in uncoordinated speech and difficulty articulating words, as we said before, interrupted speech, telegraphic speech. But in addition to all this, don't forget that Broca's area is related to proper grammar structure of the sentence, and therefore any lesion in Broca's area will result in grammatical mistakes. And of course, you know that lesions in Broca's area result in Broca's aphasia, and that the patient in Broca's aphasia does not use pronouns or small linking words. The frontal lobe is also responsible for a third function, which is voluntary motor action. And here you might wonder why we are repeating. In the previous slide, we were talking about controlling the motor actions, meaning planning for them, coordinating them. But here we are talking about the simple fact of giving a motor order to a muscle. So I think I'd better move this to be number one, because just giving an order to a muscle, this is the simplest function, but coordinating the muscles together, this is the more complex function, and we should go from the simpler to the more complex. You know here that the motor area is present, or the motor cortex is present in front of the central sulcus. Here, of course, this is the coronal section, so you cannot see you do not see the lateral aspect of the cerebral hemisphere, but this is the motor area, and you have here descending motor fibers, which we consider the upper motor neurons. The upper motor neurons arise in the motor area in the frontal lobe, descend through the internal capsule in the medulla oblongata, and then in the internal capsule in the substance of the cerebral hemispheres, and then they decussate at the level of the medulla to go to the other side and control or give motor orders to the skeletal, or actually to the skeletal muscles on the opposite side of the body. These are corticospinal fibers. So you can call them upper motor neuron in comparison to or in contrast to the lower motor neurons that arise in the spinal cord, as we have studied before. You can call them corticospinal fibers because they start in the cerebral cortex and in the spinal cord. Of course, if there is damage to that area, according, of course, to the extent of the damage, but let's imagine there is a lesion that damages the whole motor area, then there will be complete hemiplegia on the opposite side of the body, complete paralysis of the opposite side of the body. On the other hand, the parietal lobe is mainly sensory, so it receives all the somatic sensations. So what are the functions, of the broad functions of the parietal lobe? They are three functions: receiving somatic sensations, receiving proprioception, and processing all sensory stimuli to associate all this together and to help in language learning. First of all, I can ask you about lesions in the parietal lobe. So, of course, as we said with the frontal lobe, a lesion in the parietal lobe will result in failure in all these functions. You will not feel somatic sensation on the opposite side of the body. You will not feel proprioception, and therefore it would be very difficult for you to keep balance and equilibrium, and you will have difficulty, great difficulty in learning language and in math skills. First of all, what are the somatic sensations and how are they different from proprioception? The somatic sensations are sensations from the skin, muscle, and bones, like touch, pain, and temperature. And proprioception, on the other hand, is deeper sensation, and it is sensation coming from the tendons of the muscles, the joints themselves, the capsule around the joint, the ligaments. So it's deeper than the superficial, rather superficial somatic sensations. And then the third function, actually, this is number 4, the one at the bottom. The third function is interpretation of all this, understanding all this. For example, if you're touched, you have to understand that you have been touched. If an insect is crawling on the surface of your skin, this is touch. So you have to feel this. Otherwise, you will keep the insect on your skin. You have to you have to understand the position of your different joints in order to understand where you are in space and if you are in a good, safe position or you are about to fall. So all this is interpreted and perceived by, as we said before, the somatic sensory association area. And although it's called somatic sensory association area, it helps you perceive somatic sensation and proprioception. And because this association sensory area helps you understand what's going on at the sensory receptors. If it is damaged, then you will receive no information from there. You will forget about the part where the damage has occurred, and therefore this leads to body neglection or neglection of one part of the body. But it's also related, and this is function number 4, it's related to language learning, processing all stimuli related to language learning. If I want to ask you a more difficult question, I would ask you, guess what type of stimuli might be related to language learning? And first of all, of course, visual stimuli. You learn the alphabet, for example, by um, uh, learning the shapes of the letters. So you have to see in order to learn. You have to hear the sounds of those letters. So visual stimuli are important for learning, and auditory stimuli are important for learning, but also touch stimuli are important for learning because you can differentiate, for example, between a tissue made of silk and another tissue made of velvet through touch. You can never decide only through visual stimuli. So language learning, and of course, it's not only through visual stimuli, auditory stimuli, tactile stimuli, which are the touch stimuli, but through the integration of all this. So, for example, if I see a round letter and I remember its sound, which is an O, then I know that I'm talking about the letter O. But if we come to an object, for example, that you hold in your hand, in such a case, even if you are closing your eyes, through touch stimuli, you can understand what you have in your hand. But in order to understand, the association area should integrate all this. It should integrate the texture that you feel by the touch sensation, together with your previous recalls of the memory of the name of that object you're holding in your hand. And this recall is a function of other areas of the brain, like the hippocampus. The occipital lobe, as we said before, is important contains the vision center and the vision association center. The vision center helps you see what's in front of you, what's in the vision field, but the vision association area helps you understand what you see. So what happens if the vision association area is damaged? You will see the objects, you will see the faces, but you will not understand what this represents. Of course, it's important to mention that it's not only the vision association area that is important for understanding what we see, but we have to remember first what we have learned about this visual stimulus, and this, of course, comes from the fusiform gyrus in the temporal lobe. Because as you know that the fusiform virus is important for face perception, for object recognition, and also for reading. And the fusiform virus is actually present on the lower surface of the occipital lobe and the temporal lobe. So the temporal lobe helps you understand what you see in addition to the visual association area. Now, the temporal lobe, of course, is very important because in the temporal lobe, we have the auditory area, we have the auditory association area, we have vernix area. And as you know, vernix area is responsible for producing ideas of speech, for understanding the meanings of words. And if vernix area is damaged, then you will have vernix aphasia. And vernix aphasia is sensory aphasia because people with vernix aphasia articulate words very well. They do not have any problem with motor functions, but they give fluent speech, but nonsense speech because they cannot produce meaningful speech since they do not essentially understand the names or understand the meanings of the words. And in vernix aphasia, the patient is not frustrated because he does not understand that he has a problem. So first of all, if the vernix area is damaged, then vernix aphasia will occur. The person would not understand language. The person would not be able to generate or formulate ideas of speech, and he would not be able to express himself well, and he would have very fluent speech. Of course, this occurs if vernix area only is damaged or if the whole temporal area is damaged. If the auditory area is damaged, you do not hear the sound. If the auditory association area is damaged, you hear the sound, but you do not understand it. And again, we have sequencing and organization. We have seen sequencing and organization in the frontal lobe, as well as in the temporal lobe, as well as in the parietal lobe. So they all help this function. As we said before, the nervous tissue is essentially made of neurons, which are these nerve cells, whether they are sensory nerve cells or motor nerve cells. But it's also made of other cells that are called the neuroglia, as well as of blood vessels. So the neurons are the main cells in the nervous tissue. But in addition to the neurons, we have the neuroglia, and we have the blood vessels. We previously talked about the functions of the neurons. They receive impulses through dendrites. The axon transmits the impulses, and then they synapse with another neuron or a gland or a muscle. And the synapse, how do we call the synapse between a neuron and a muscle? The synapse between a neuron and a muscle is called the myoneural junction. We have different types of neuroglia cells. As you can see here, we have the neuron, which is the main type of cells in the nervous tissue, and we have blood vessels, and we can see three types of neuroglia cells. The first type of neuroglia cells to the left are the microglia cells, and they are responsible for local immune function in the brain. And then we have the astrocytes, and they are so called because the prefix astro means star. They are star-shaped, and as you can see, they have podocyte. They are called podocytes because they have feet. They are podo means foot and cyte means cell. So the astrocytes are considered podocytes because they have foot processes. And as you can see, the astrocyte stretches itself between the cell body of the neuron that requires the greatest amount of nutrition and the blood vessels. So the food processes absorb the nutrients and oxygen and everything from the blood vessel and transports it again through another food process on the other side to the cell body. The last cell in the neuroglia cell are the last type of cells are the oligodendrocytes, and the oligo means few. So they have few dendrites, oligodendrocytes in the brain, in the central nervous system, are the equivalent of Schwann cells in the peripheral nervous system. They are mainly used to form myelin sheath around the axons, but they are also important for repair whenever damage occurs to the axons. In general, if you're asked about the function of neuroglia cells in general, you would say that they provide neurons with glucose and oxygen via their podocytes, and this is mainly the function of the astrocyte. They insulate the neurons, partly because they form the myelin sheath, which is an insulator, and partly because they themselves are considered insulators since they do not transfer any type of electricity. They support the neurons just by being there, and they anatomically connect the neurons, as you can see, and they help healing and repair. Of course, Schwann cell is present in the peripheral nervous system mainly, so we would say oligodendrocyte here and there instead of the Schwann cell. So they help healing and repair, and they help formation of the myelin sheath. We talked about the myelin sheath in the previous video, so we are not going to repeat it. But if you get this diagram, for example, and I might ask you about the process, you would say that it's saltatory conduction. It's a rapid type of conduction that occurs in myelinated axons because of the presence of, it's not enough to say because of the presence of the myelin sheath, because if the myelin sheath were continuous, we wouldn't see saltatory conduction. The main idea is that the myelin sheath is deposited in the form of interrupted segments. So in between the segments, we have non-myelinated parts. So I might ask you to label the non-myelinated parts. So here, the yellow parts are the non-myelinated parts, and you can see that the electrons move from one node of Ranvier to the other. How do we call the non-myelinated parts? We call them nodes of Ranvier. What do these nuclei represent? They represent the nuclei of Schwann cells, or the nuclei of the oligodendrocytes. And here, of course, if I ask you about the diagram, you would say that at the beginning, the Schwann cell stretches itself around the axon. The yellow circle here is the cross section in the axon, and the blue crescent here is the Schwann cell with its nucleus, or the oligodendrocyte. And it has just formed one turn, and then after time passes, we have several whorls or several turns. So these concentric layers, concentric means having one center, which is the axon. These concentric layers of cell membranes or phospholipid form the myelin sheath. The synapse, of course, is very important. The synapse is the area where the terminal arborizations of one neuron come very close to another neuron, or to muscle fibers, or to a gland. Muscle fibers and glands are called effector organs, okay? Of course, as you know, the neurons can be sensory or motor. If a neuron receives impulses, if a neuron carries impulses to the central nervous system, then it is an afferent neuron. If it carries impulses away from the central nervous system, then it is an efferent neuron. So we consider the neurons afferent and sensory, or efferent and motor, according to whether they transfer towards the central nervous system or they transfer away from the central nervous system. For example, here in this reflex action, whose center is the spinal cord, that neuron is carrying impulses towards the spinal cord, then it is a sensory afferent neuron, whereas that neuron is sending impulses away from the central nervous system or the spinal cord, then it is an efferent motor neuron. And it is called an efferent motor neuron, whether it is going to a skeletal muscle or to a smooth muscle or to a gland, okay? This is called a motor unit. A motor unit is the nerve axon and its branches and all the muscle fibers that are related to it, and they contract as one unit when the electric impulses reach this terminal part of the axon, a motor unit. Now, this slide is very important and we explained it a lot. Here we have a typical motor neuron, and you can see how the impulses are going from the cell body towards the terminal arborization. So a typical motor neuron has a cell body, dendrites, a long axon, and at the end you can see the effector organ. So the impulses are received at the dendrites and they're sent along the axon to reach this synapse, which is called a motor end plate or a myoneural junction or a neuromuscular junction. Actually, motor end plate is not the name of the synapse. The name of the synapse is neuromuscular junction or myoneural junction, the junction between a muscle and a neuron. But motor end plate is this thickened part, or not actually thickened only, but this modified part of the muscle cell membrane that facilitates the entry of sodium and potassium, and therefore the motor end plate is part actually of the cell membrane of the muscle. On the other hand, the sensory neuron is very different from the typical motor neuron. Here, the sensory neuron is myelinated, but most sensory neurons are not myelinated. The sensory neuron has a different shape. It has like a head, a ganglion, a cell body, and that cell body does not have the typical dendrites. And it has a short process that is then divided into a central branch that goes towards the central nervous system, for example, the spinal cord, and a peripheral branch or division that goes away from the central nervous system. However, whether we are talking about the central branch, which is towards the central nervous system, or the peripheral branch that is away from the central nervous system, the impulse direction is the same. Notice that there is a receptor here, or the receptor actually is the modified sensory nerve ending, and starting at the receptor that transduces what is a receptor. A receptor, this is very important, and you always make mistakes when defining a sensory receptor. A sensory receptor is a modified sensory nerve ending that is capable of transducing any type of energy into electric energy. And the electric energy in the sensory neuron moves along the axon, along the distal branch, and then, or the peripheral branch, and then along the central branch until we reach the spinal cord. So whether, if I ask you, for example, to draw arrows to represent the direction of electric impulse transmission, then it would be always towards the spinal cord and away from the receptor. On the other hand, in the motor neuron, it's in the opposite direction. Here, as we said before, the spinal nerve, we have 31 pairs of spinal nerve, one here and one there, and we have 31 of these. Every spinal nerve is a mixed peripheral nerve because it consists of sensory fibers and motor fibers that are connected together inside the nerve membrane or the nerve sheath. But as the mixed spinal nerve approaches the central nervous system, it divides into two roots, a dorsal root and a ventral root. Of course, if I ask you about the difference between the dorsal root and the ventral root, the dorsal root is posterior and the ventral root is anterior. That's why this one is called dorsal and that one is called ventral. The dorsal root is sensory, purely sensory, and the motor or the ventral root is purely motor. So this tells you what? It tells you that as the spinal nerve approaches the spinal cord, it divides into two branches, one of them being purely sensory, one of them being purely motor. So they mix together only inside the spinal nerve, but outside the spinal nerve, the dorsal root is purely sensory and the motor root is purely motor. The ventral root is purely motor. In between them, you might or not have an interneuron. An interneuron transfers the impulses from the dorsal sensory root to the ventral motor root. The presence of the interneuron delays the reflex a bit, meaning that if you need the reflex to be very rapid, like the withdrawal reflex, for example, when you are pinpricked, you withdraw your hand, then you wouldn't have this interneuron. But the presence of the interneuron, although it delays a bit the reflex action, yet it is very important because it allows this reflex arc or this pathway of the reflex action to synapse with other neurons. For example, to synapse with a neuron that crosses to the opposite side and then ascends to the brain. Of course, what does this diagram represent? It represents a reflex action. What is the center of the reflex action here? It is the spinal cord. Do all reflexes have their centers in the spinal cord? No. Most of them have their reflexes in the spinal cord because the spinal cord is very long, but there are reflexes like the pupillary reflex, for example, that have their centers in the brain or in the brain stem. The reflex action is an automatic involuntary action whose purpose is to protect the body. What are the components of the reflex action? We have a receptor, we have a mixed spinal nerve, we have a sensory afferent neuron, we have an interneuron, a ventral motor neuron, and then an effector organ. The structure of the spinal cord, you have an inner shaped H-gray matter. And what is gray matter in the spinal cord? It consists of cell bodies of neurons, it consists of dendrites of neurons, and it consists of the unmyelinated parts of the axons. Around it, we have the white matter, which is essentially formed of myelinated axons in the spinal cord. In the brain, it's the same. The gray matter is made of nerve cell bodies, dendrites, and a very small portion of unmyelinated axons, and the myelinated, the white matter of the brain is made of myelinated fibers. But the difference is that here the gray cortex, the gray matter is inner or central, whereas in the brain, the gray matter is outer, and the gray matter in the brain is responsible for the highest and most complex functions in the body. The sensory functions of the nervous system arise in the sensory receptors. As we said before, what is a receptor? It is a modified sensory nerve ending that can transduce any type of energy into electric energy. Here we have examples of receptors. So you have to know them, of course. You have the Meissner's corpuscle, which is responsible for light touch, and therefore it's very superficial. Of course, you can see that all the receptors lie in the dermis, in the connective tissue layer of the skin, not in the upper epidermis. So you have the Meissner's corpuscle for touch, and on the other hand, the one that is very deep, you have the Pacinian corpuscle for pressure. Pacinian starts by P, and pressure starts by P. You have these modified sensory nerve endings that look like a tree that are the nociceptors. Something noxious is something harmful, and the nociceptors are responsible for detecting pain, and the thermoreceptors are responsible for detecting changes in temperature. We have two types. We have Krauss bulb, Kraus, that is responsible for cold, K, cold, Krauss bulb, and we have Ruffini end organ. They do not show here. Ruffini is for heat, Ruffini. Of course, all these sensations are somatic sensations. So temperature, touch, pain, and pressure are somatic sensations, and they are detected in the primary sensory cortex of the brain. So the receptor transduces energy into, for example, heat energy into electric impulses that will ascend to reach the parietal lobe. So their final destination is the parietal lobe. If I ever ask you about the representation of the body in the motor area or in the somatic sensory area, you would say that, first of all, there is an upside down representation, meaning that the face is lower down and the legs are upper. And on the other hand, there is something else, which is the disproportionately large area that is dedicated to the face, to the mouth, to the lips, and to the hands. Concerning, if I ask you why, of course, you have to separate the sensory from the motor. Sensory, this means what? If it's represented by a large area in the somatic sensory area, this means that there is a large number of sensory cells that receive impulses from a large number of receptors on the face and then integrate all this, which means that we will have more accurate and more precise sensation. On the other hand, what does this mean for the motor area? When you have a large motor area that represents the face, this means that you have more motor neurons that control the muscle of the face. This means a more precise and accurate control of the muscles of the face. And this makes sense because the muscles of the face are multiple, they are small, they carry out very specific functions in order to show the complex facial expressions, and therefore they have to be supplied by a large number of motor neurons. And then we come to the last part of the part included in the mid-semester test, which is the sensory function of the nervous system. Of course, here we have the optic nerve, the pathway of the optic nerve. So the optic nerve, here this is the optic disc, and the optic nerve starts here at the optic disc, and then it leaves the orbit, or here, of course, you know that the skull has a bony, a bony depression to accommodate the eye, and the optic nerve leaves here through a foramen or a hole in order to reach the vision association area in the vision area first in the occipital lobe of the brain. And I'll be showing you the Word document, sharing it very soon, in order to tell you that the optic nerve starts at the retinal ganglion cells. So the optic nerve is nothing but the axons of the retinal ganglion cells. Here we can see the retina, and the retina is a delicate nervous tissue that coats the posterior part of the eye, and the retina is made of nerve cells, and the most important nerve cells are the rods and the cones. The rods and the cones are, we do not see them here, of course, and you will not see them upon ophthalmoscopic examination of the retina, but you can see them in the diagram. So I'll be talking rapidly about them until I show you the diagram. So they are photoreceptors. They can transduce light energy into electric energy or electric impulses that pass along the optic nerve. The rods contain one type of pigment, which is rhodopsin, rhodopsin, and it is responsible for dim light vision. And since it is dim light vision, then you see only shades of gray. And the rods, since they can see in dim light, then they are highly sensitive to light. So they have high sensitivity to light, but they cannot give you accuracy of vision because it's dim light vision. On the other hand, the other type, which are the cones, which are conical in shape, have three types of pigments. And these three types of pigments merge together to give you all the shades of colors you know. And the cones can give you more accurate, more precise, and color vision. Let's come to the retina and the ophthalmoscopic examination of the retina. What can you see here? You can see the optic disc, which is the structure where the optic nerve arises. in the retina and then leaves the eye or leaves the eye to go to the occipital lobe and reach the vision area. And you can see the fovea, and the fovea is the part of the retina where you have where it's needed for the light rays to converge in order to see accurately. If the light rays do not converge on the fovea, then your vision will be blurred, and you can see blood vessels. And these blood vessels are important for nutrition of the nerve cells of the retina and providing them with oxygen and glucose. If you examine the retina, you have to, there should be regular checkups of the retina, especially in people who have diabetes or hypertension, because there the blood vessels of the retina might become damaged or narrowed with diminished supply to the retina of oxygen and nutrients, resulting in retinopathy and damage to the cells of the retina and maybe blindness or loss of vision. We call this retinopathy when the retina is sick or diseased because of the microvasculature of the retina that is damaged. And of course, ophthalmologists and doctors that can understand the changes can detect these changes. But you can also see traumatic changes to the retina, like small retinal tears or even retinal detachment. When the retina is detached, it's undulant, it looks like waves. It's no more flat like this. And how does this affect the patient? If the patient sees opacities, or if the patient sees floaters, or if the patient sees like a curtain shadow in front of the eye, then this is, of course, an indication, especially the curtain or the wedge-shaped opacities are an indication of retinal tear, and the wedge-shaped opacities indicate retinal tear, and the curtain shadow indicates retinal detachment. Other than this, in myopes, the retina is stretched out because their eye globe is oblong, and in such a case, they would see floaters and they would see flashes of light. If you see flashes of light at the periphery of your visual field, especially in dim light, this is an indication of not an actual retinal detachment, but a pending retinal detachment. Another function of the nervous system is the detection of the auditory stimuli. And as you can see, we have here inside the inner ear you have two important structures. You have the cochlea and you have the semicircular canal. The cochlea is the organ of hearing, and the semicircular canals are the organ of equilibrium. The cochlea is the organ of hearing because it contains fluid and it contains hair cells inside this spiral structure. So and there is a very thin membrane here that is affected by the three bones in the middle ear. So when sound enters and vibrates the eardrum or causes vibration of the eardrum, the malleus, the incus, and the stapes, which are the three bones in the middle ear, will vibrate, and they will cause vibration of the oval membrane in the cochlea, and this vibration will result in vibration of the fluid inside the cochlea and the hair cells, and this vibration will be transduced into electric stimuli that pass along the auditory nerve. And the auditory nerve relays here in the ventral and dorsal cochlear nuclei, and then in the superior olive in the pons varolai. And then there is an ascending pathway from the ventral nucleus on the same side and from the dorsal nucleus crossing to the opposite side. So this is very useful because sound stimuli will be distributed on the two ears, and this is very important just in case damage occurs in one pathway, the other pathway would be functioning from the same ear. And it's also important because higher functions can detect the site of the stimulus. We have another synapse or relay in the inferior colliculus in the midbrain, and then in the medial geniculate nucleus and then to the auditory cortex. Of course, I'll never ask you about the nuclei and their names unless I get you the diagram. If I don't get you the diagram, then you talk only about the auditory nerve that carries the impulses to the auditory cortex. And the auditory cortex, as you know, is in the temporal lobe. Concerning the olfactory stimuli, any odoriferous substance or volatile substance will stimulate the sensory receptors here, which are the terminal branches of the olfactory nerve. And then the axons of these cells will pass through perforations or holes in this bone that is called the ethmoid bone. The ethmoid bone in the area where it is perforated is called the cribriform plate. And then the cells will relay here in the olfactory bulb, and then they will form the olfactory tract on the lower surface of the frontal lobe until they reach the superior temporal gyrus until they reach the medial aspect of the temporal lobe in the olfactory area. You can see here, of course, the olfactory area that is on the medial surface of the temporal lobe. Concerning taste sensation, the taste receptors from the anterior two-thirds of the tongue are carried by the facial nerve, whereas taste sensation from the posterior third is carried by the ninth cranial nerve, which is the glosso referring to the tongue, pharyngeal nerve, and then they ascend, pass through the medulla oblongata on the same side, and then through the thalamus, where all sensations are relayed, and then to the gustatory cortex in the insula, as you know. So, of course, if the facial nerve is damaged, you can lose taste sensation from the anterior two-thirds of the tongue. Here we have the vestibular system. As we said, we have the cochlea in the middle ear, but in the inner ear, but we also have the semicircular canals. So the semicircular canals do not show here, although it is a slide that is talking about balance, but the pathway for balance starts here at the semicircular canal, which should show here. And then, as you know, where your position changes, there are impulses that pass along the vestibular division of the eighth cranial nerve, relays in the cochlear nucleus, and then ascends to the brain to tell the brain about your position. But here, of course, as I said before, unfortunately, I haven't changed the diagram. This diagram is not the most suitable one because the vestibular branch and the vestibular sensations should reach the cerebellum, and the cerebellum will give impulses to the spinal cord, cerebellospinal impulses to make the anti-gravity muscles correct your position. So if this were the proper diagram, then we should see here the vestibular, we should see here the semicircular canal, but the proper diagram is present on the document. I might ask you about the two divisions of the eighth cranial nerve. So you have the auditory division of the eighth cranial nerve, which comes from the cochlea, and the cochlea is therefore the organ of hearing. And we have the vestibular branch, which comes from the semicircular canals, which is the organ of equilibrium, and therefore the vestibulocochlear nerve, or the eighth cranial nerve, is responsible both for hearing and for equilibrium. Okay, I'll stop here and then continue the third part later on.